Hey learners, welcome and welcome back to the part 5 of this chapter 8, Cell the Unit of Life. Topics to be covered in this chapter are ribosome, cytoskeleton elements and their types, and at the end if possible we'll also try to cover nucleus. So let's begin by the chapter. As we all know that ribosomes are the protein factory of the cell, but where did it came in notice and how? So let me take you through their discovery first. So the ribosomes came in notice in 1950s when the electron microscopes was discovered. But before that, scientists already suspected that there is something proteinaceous were made in the cytoplasm. But in the 1950s, when the George Palade observed some small dense particles attached to that endoplasmic reticulums by some electron microscopes, he called these particles as Palade particles. Later on, these dense granular particles were named as ribosomes, where ribo refers to the ribosomes, which is a sugar found in the RNA, ribonucleic acids, and the zome means the body. It means they are like the RNA body or RNA containing structures. This makes sense, right? Because the ribosomes are composed of ribosomal RNA or in short rRNA and protein structures. That is why they are also called as the ribonucleoproteins. If we talk about their size, the ribosomal size is confirmed in nanometers, which is approximately about 20 to 30 nanometers. That is why ribosomes are considered as the smallest cell organelles. And because of their small size, a single human cell can have millions of ribosomes in them. Despite of their size, they play one of the most important role in protein making. So they are known as the protein factory of the cell, and they are considered as the universal cell organelles as they are present in both the prokaryotes and eukaryotic cells as a one organelle within another organelle, except the human mammalian RBC. Other than all the living cells have ribosomes in them. Ribosomes are also very unique among all the cell organelles in terms of their structure, as they are non-membrane bound organelles. It means they are not surrounded by any membrane or any structure, so they are considered as the membrane-less organelle, unlike most of the other organelles in the eukaryotic cells. Now tell me why they are called as the engine of the cell. See, just like an engine powers a car or any mechanical tool by burning fuel and creating motion to it, similarly the ribosomes power the cell by making proteins in it. You can imagine by your own. Think of a car. It can still roll without the tires, or maybe it can coast on the slides without the fuel for a moment. But without an engine, the car simply cannot run. So the ribosomes are like the engine of the car. So without the ribosomes, no protein would be made, and without the proteins, life simply wouldn't exist. Even the medicines like antibiotics takes the advantage of it, like tetracycline or erythromycin, if you know, we used it in COVID. So they work by targeting bacterial ribosome and shutting down their protein production. This kills the bacteria without harming human cells, because our ribosomes are slightly different from them. Now let's see their structures in detail in the next slide. What are the types of ribosomes? So first we have prokaryotic 70S ribosome or eukaryotic 80S ribosomes. If you remember, in the first part of this lecture we have already done by the prokaryotic 70S ribosomes, but in this part we will try to cover from a basic to an advanced level so that you can easily differentiate between these two ribosomes at any time. Ribosomes can be found in two different ways in the cell. They can be fixed or they can also be freely found. So when the ribosomes are fixed to the cell surface, or in the second case, they can also be freely found in the cytoplasm of the cell. So let's see it on both the cell types. So in the eukaryotes, the 80S ribosomes occurs in the cytoplasm of the eukaryotic cells in the free form, or they can be also associated with the endoplasmic reticulum or nucleus of the eukaryotic cells in the fixed form. But in the prokaryotes, the 70S ribosomes occurs only in the free form, as they are associated with the surface of plasma membrane of the prokaryotic cells, as they don't have any membrane bound cell organelles within them. They are not found fixed like the ATS eukaryotic ribosomes does. By the previous lectures, you already know that the 70S ribosomes are present in the prokaryotic cells and the ATS ribosomes are present in the eukaryotic cells. But the 70S ribosomes are not just only present in the prokaryotic cells, while they are also present in other cell organelles of the eukaryotic cells like the chloroplast or the mitochondria, which you need to remember. Why so? As they have the bacterial features in them, correct? But the ATS are only present in only the eukaryotic cells. If we talk about the structure of ribosomes, they are composed of two major subunits. The first is the smaller subunit and the another one is the larger subunit. When these subunits combines together, then only ribosomes can form protein and can behave like the protein factory of the cell. The assembly of both the ribosomal subunits take place only when the concentration of Mg+2 ions is high in the cytoplasm of the cell. You just can't call them smaller subunit or larger subunits every time. They have their own specific name, which is also important. So their name is different in both the 70S and 80S ribosomes. Let's see what are these. So the 70S ribosomes are composed of two subunits. One is the smaller subunit, which is 30S, and the larger subunit, which is 50S. You can remember it by, as you can see, the 7 is an odd number and the 30 and 50, the starting of their 3 and 5 both are the odd numbers. So you can remember it like this, and don't try to add 30 plus 50 80, because the even time sum of two odd digit number will always be even, and 80 is itself an even number. That is why I'm saying, like, don't use your extra 3 brain cells here. That I know that is mathematically correct, but that contradicts it while adding. Then in the ATS ribosomes, they are again composed of two smaller or larger subunits. Smaller one is 40S and larger one is the 60S, where 8 is an even number in the interval of 10, which you can see in their subunits too. Okay, wait, let me repeat it again for you. So this is a very simple trick, I guess, where the odd ribosomes is made up of the odd subunits, such as 70S ribosomes, where 7 is an odd number, is formed by the association of 50S and 30S subunits, where 5 and 3 are the odd numbers. Similarly, this can be seen in the ATS ribosomes, where 8 is an even number and it is formed up of 60 and 40S subunits, where 6 and 4 are even numbers again respectively. You can use this mnemonics or you can also create your own, and if you can remember it raw, then also very well and good. In the 70S and 80S subunits, how come their addition of 50S and 30S will be 70S and similar to that of 60S and 40S will be 80S? What are these and where these S came from and what does it mean? In the first impression, it sounds like a measuring size or unit, but actually it is not. It is not even the addition, it is just a measurement. It's a common point of confusion among all the students. So let's see what are these. The S stands for the Sedgwick unit or the sedimentation coefficient rate, which indirectly measures the density and sizes of how fast a particle sediments or settle down. Let's see this how the inactive measurement actually works in the practical point of view. Like, imagine you have two balls, one is a solid metal ball and other is a hollow plastic ball of the same size. So when you throw a ball to your friend, you don't actually measure the ball's weight directly. You get an idea of how heavy or light it is by how far and fast you can throw it or how hard it is to throw. The plastic balls travels much faster than the solid metal ball. Why so? The plastic ball is lighter, but it just not depend on its weight, but also its shape and how you throw it. Similarly, in the world of molecular biology or tiny particles like the ribosomes, we cannot see their size directly under a microscope, nor we can just directly measure the size or mass of that particle. Instead, our great volunteers or scientists introduces a centrifugal technique where the centrifuge machine is used. It is basically a super fast spinning machine which is mostly used in the laboratories to separate plasma from the whole blood and to separate sediments from the urine. But in this lecture, we will use it to see how fast the ribosomes sedimentates. This experiment works on the principle of centrifugal force in physics, although you must have seen this experiment in your junior classes. If not, then anyways we'll talk about it in this lecture. So let's see what are the procedures. So for that, first you need to take the ribosome samples isolated from the cell, then load your sample into the centrifuge tube suspended in a buffer solution. Why we using this buffer solution? As the word buffer means to balance. This is a liquid that keeps the pH stable and balanced. It doesn't even react with any other cell organelles or interact with any of their bonds, and it keeps the solutions separated within the tube. Look, the buffer solution is just a mixture of weak acid or a conjugate base, and similarly you can take a weak base or conjugate acids. These components can neutralize any added acidic or base by preventing the large shifts in its solution's pH, which is crucial for many other biological activities and chemical processes also. As I said, you can't even use water in the place of buffer solution, as their H2O molecules can mix with the cell's aqueous environment. Now you know why the buffer solution is considered as the ideal solution, as it doesn't even have any water molecules in it. Now in the step 2, set up a sucrose density gradient. For that, you have to fill the test tube with the layer of sugar solutions, which is also called as the sugar gradient. You will see that the sugar concentration is low at the top and high at the bottom, forming a gradient in it. This actually helps to separate the particle of different sizes or densities to settle at a specific point in the gradient. You can even see in the diagram that the layers of sucrose mixed with the buffer solutions to create a thick syrup-like effect that separates the ribosomes based on their size, mass and shape. In the step 3, the ribosome solution is carefully placed on the top of the sugar gradient in the test tube. Now in the step 4, place these tubes in the rotor, ensuring they are balanced by adding an equal volume of water to the opposite tubes. It is necessary to balance the load in the machine for their proper functioning. So for that, if you have an odd number of samples, add a tube filled with water to the same volume as the sample tubes, and place that tubes directly opposite to each other in the router, or simply you can just keep all the tubes along with your sample tube in the router. In the step 4, close the lid, set the speed and time on the controllers and start the cycle. The centrifuge spins at the very high RPM rate. The spinning forces the ribosome molecules to move through the sugar layers on their mass and density. Once the cycle gets complete, wait for the rotor to stop, then carefully remove your separate samples from the centrifuge machine. After some time, you can watch the ribosomes stop at different positions in the gradient. The bigger and heavier ribosomes sink further, and the smaller or lighter ones stay higher up. This is the after spinning diagram of the test tube. You can clearly examine and can measure the results by your own, not by putting it on a scale, but instead we observe how it behaves in the spinning environment and can calculate the S value of their weight and density based on its movement. That the SSU smaller subunits with the 18S or 16S rRNA types, where 18S is in the eukaryotes or 16S is in the prokaryotes, sediments less because they are smaller and lighter, higher in the gradient. You can see their location in the tube. It lies in the middle layer or above the ribosomal palate, or in the larger subunit LSU with the 20S or 5.8S and 5S lies in the eukaryotes, and the 23S or 5S rRNA types lies in the prokaryotes. The sediments further down and forms a lower gradient. You can see in the test tube that they just lies above the ribosomal palate. Both subunits together even goes further down, eventually forming a palate at the bottom, as you can see in the diagram. You can think of it as a swimming pool. The lighter people or smaller subunits float near the surface, or the heavier people or the larger subunits are super heavy people, so they can sink all the way at the bottom. So the rate or speed at which the rRNA molecules settle down or sediments, that rate is called as the sediment coefficient rate and the Sedgwick units. This centrifuge machine is also used to check the purity of the milk. You can check it out in your nearby dairy, where the fat creams, being the least dense components, rises on the top of the tube, and the skimmed milk, which is mostly the water, proteins or minerals, forms the middle layer, and whatever the impurities, sediments added in the water that affects the thickness of the layer can be detected because they forms a separate layer in it. Now let's see what are the types of rRNA or proteins present in the ribosomes in a little more detail by the next slide. As we all know that ribosomes are the rRNA bodies, and this diagram it particularly shows the different types of ribosomal RNA and the ribosomal proteins involving in the formation of ribosomal subunits, where these distinct molecules plays the different structural and functional roles in the ribosomes, where both of these prokaryotic and eukaryotic ribosomes consist of a specific rRNA type and dozens of proteins in it that works together to make the life's essential proteins out of it. So let's see what are these. In the first panel, the prokaryotic ribosome is given, where the 50S rRNA components in their larger subunits are 23S rRNA and 5S rRNA. And in the 30S smaller subunit, it is 16S rRNA, where around 21 proteins in the smaller subunit and 31 proteins in the larger subunit helps to stabilize the structure of the ribosomes along with the different rRNAs in it. And in the bottom panel you can see the eukaryotic ribosomes, where the rRNA composition in the larger 60S subunit is 20S rRNA and 5.8S rRNA and 5S rRNA, and in the smaller 40S subunit it is 18S rRNA in it. And if we talk about their protein components, it has 50 proteins in the larger subunit and 33 proteins in the smaller subunit, where these 2900 R NTS and 120 R NTS refers to the length of the rRNA strand. We know that these nucleotides are the basic building block elements of these RNA or DNA compounds, where each nucleotide consists of sugar, phosphate groups and nitrogen bases. But you don't need to know all these things for now. We will discuss them in some other chapter. And yeah, one more thing I would like to clear here, that many students simply tries to add the ribosomal proteins together like 31 plus 21. But you should not. Hear me out. You should not add 31 plus 21 because that is not equal to 52. Here just to describe the total number of ribosomal proteins. No, you cannot do that because there is an overlap, and how we count them, that actually matters. But if you really like to contradict my point and try to add them, you can give it a try. Like if you add 31 plus 21, it will be 52. As soon as you write 52, your complete answer will be wrong. See, the total number of ribosomal proteins in the complete prokaryotic 70S ribosome by combining the both smaller and larger subunits is 54 proteins, not the 52. Yes, you heard me right. And in the eukaryotic ribosome, the total number of proteins will be 80 proteins in them. Let's come to the point. Why not we just add them? What is wrong with it? See, because some proteins might have homologous in both the subunits, but they are counted separately when describing larger versus smaller subunits in them. So the 54 ribosomal proteins in the prokaryotes and 80 ribosomal proteins in the eukaryotes are generally accepted numbers. Likewise, we never add Sedgwick units together. For example, 23S or 5S are RNAs because they are not the additive measurements. We know that. Instead, we treat them as individual components of the ribosomal subunits, where each comes with their own specific role in them. And the distinct molecules refers to a single rRNA species, not just the sum of the smaller rRNAs. See, each number is unique and holds some meaning in them, so it is better to remember them as it is. And I would suggest you not to mix and match these numbers with each other. That would be better for you. And if any one of you student is confused in the structural position of ribosomal subunit, then let me clear you this, that the position of smaller and larger subunits is not fixed. In an absolute sense, like the always upper or lower, or maybe the always lower or upper, but instead their relative positions depends on the perspective in which the ribosome is visualized by you only. In most of the scientific diagrams and models, the smaller subunit 30S in prokaryotes or 40S in eukaryotes is often depicted on the top side or the upper side, and the larger subunit 50S in prokaryotes or 60S in eukaryotes is depicted on the bottom or the lower side of the ribosomes. This all conventions is used for the clarity, especially this visual helps the learner to initiate and to know how the mRNA actually enters the smaller subunit and how these peptide bonds exits from the larger subunits. So this is all for the learner's point of view and their understanding. So that matters biologically is the correct alignment of the mRNA, tRNA and the active sites in the ribosomes, not a fixed orientation. Don't take stress about it. We will see these mRNA and their functioning in the next slide. So there is no universal fixed up or down part in the cell. The subunits associate based on only their functional requirements. So this is your task for today. You have to remember the types of RNAs and the proteins, or you can avoid the nucleotide composition for now. Now let's just move by the next slide. Then we have salt linkage or the salt bridge, which is a very important concept of organic chemistry, and it also becomes important in the terms of biology when it comes to ribosomes. So let's see what are these. So the salt linkage is a type of non-covalent interaction or bond that occurs between the oppositely charged groups in the biological molecules, such as in the ribosomes it is the protein or the nucleic acids. So as I said in the previous slides that this is your larger subunit and this is the smaller subunit of your ribosomes, and these two subunits will be assembled only when the salt linkage will take place with the help of Mg+2 in it. This question has been asked multiple times in the exams, that which element plays the key role in the protein synthesis, and the answer is Mg+2 magnesium ions. See, these magnesium ions are essential, very essential for the binding of the ribosomal subunits in the salt linkage. Actually, the Mg+2 helps to form the ionic bond with the negatively charged phosphate groups of the rRNA interacts with the positively charged amino acid of the proteins to assemble both the subunits together on the mRNA into a functional ribosome, which is also called as polysome. Now let's see what are these polysome structures. And yeah, one more thing, the concentration of Mg+2 should always be higher in the cytoplasm of the cell. The minimum required concentration is 0.001 molar is required for structural formation of ribosomes. Below this concentration, the ribosomal subunits can be unstable and even they can tend to dissociate or separate at the low concentration. Let's understand this by their structures. So you may have probably heard about the central dogma of the molecular biology, which explains how genetic information flows inside a cell. Inside the nucleus in the eukaryotes or the cytoplasm in the prokaryotes, the DNA sequence of a gene is copied into the messenger RNA or mRNA by the process called transcription. To convert the mRNA into the proteins by the process of translation, we need some ribosomes. So both the subunits of ribosomes comes and binds with the structure of mRNA. So in the simple terms, you can say the mRNA acts like a blueprint carrying the genetic code from the DNA to the ribosomes, where these ribosomes reads the mRNA sequence in the set of 3 nucleotides called codon, where each codon codes for a specific amino acid. But that is for the next chapter, right? In this part, let's talk about what is going inside the ribosomes first. Now you know how important these ribosomes are. Even without these ribosomes, the central dogma would stop at mRNA only. So, no protein, no life. Now, let's see what are the other important features of the protein in the next slide. Now, we have the term called polyribosome or polysome in short. So, the several ribosomes may attach to a single mRNA and forms a chain called the polyribosome or polysome in short. And then these ribosomes of the polysome translates the mRNA into the proteins. So let's see what are the steps required for the assembly of ribosomal subunits. So in the step 1, the smaller ribosomal subunit 30S in the prokaryotes or 40S in the eukaryotes binds to the mRNA strand. Then in the step 2, the joining of larger subunits 50S in the prokaryotes and 60S in the eukaryotes takes place. As a result, the fully formed functional ribosomes are assembled now. Then in the step 3, the tRNA molecules bring amino acid corresponding to each codon, where each codon codes for a specific amino acid. Now in the step 4, finally, the ribosome catalyzes the formation of peptide bonds between the amino acids. As a result, the fully formed functional ribosomes are assembled now. Now the ribosome is ready to proceed with the elongation, reading the mRNA codon and adding the amino acids to build the polypeptide chains so that the growing protein chains emerge from the ribosomes at the end. Once finished, the protein is folded into its shape and ready to work. Since there are more than one number of ribosomes on its structure, it is called as polyribosomes. Very important from their exam point of view. That is all about it. You will see this complete protein mechanism in detail in class 12th of the chapter called Molecular Basis of Inheritance. Now let's move by the next slide. Perfect. Now just quickly summarize all this together in a brief differentiation between all of these 270S and 80S ribosomes. No doubt that the eukaryotic ribosome is more complex than the prokaryotic ribosome, both in their size and structure. You can clearly see even in the diagram also. Their sizes varies. Their size is 15 to 20 nanometers in the 70S and 25 to 30 nanometers in the 80S ribosomes. The 70S are present only in the free form associated with the plasma membrane, while the 80S ribosomes present in both free and fixed form. And you can see their structural composition. The prokaryotic ribosome relies more on rRNA for their catalytic functions, while the eukaryotic ribosome have more proteins in them for their structural complexity. You can see the prokaryotes are composed of 60% rRNA while only 40% proteins are there, and vice versa the eukaryotes has 40% rRNA and 60% protein composition in it. So overall you can say the ribosomes are composed of half protein and half rRNA in it in their structural composition. By combining their both the subunits 50S and 30S, it will be 70S in prokaryotes, and 60S and 40S, it will become 80S in the eukaryotes. But instead of all these differences, ribosomes are almostly identical across all the life forms, from bacteria even up to humans. That means that evolution has kept them nearly unchanged for billions of years. So it is that important. Now let's move by the next slide. Now we have the cytoskeleton elements, where the name cyto means the cell or the cytoplasm of the cell, and skeletal means the skeleton system in our body which provides the structural framework to our body. Similarly, the cytoskeleton elements provide the structural support to the cell. Or you can also compare it with the steel beam in a building. Just like the steel beams hold up a building, similarly the cytoskeletons hold up the cell from the inside. It is an elaborated network of filamentous proteinaceous structure. It is a protein-based structure which is present in the cytoplasm of the cell. It is present in all the eukaryotes, both in the plants and animal cells, except the prokaryotes. Prokaryotes lacks these cytoskeleton elements. They don't have as such membrane-bound organelles in them, and the rest of the organelles are freely floating in their cytosol, so they don't need this complex structure in them. These are few of the functions which is given in your NCERT. So the cytoskeleton elements helps in the mechanical support, as I said, and they are involved in the maintenance of the cell shape and involved in the mortality of the cilia and flagella, which we will see in the next slide. And these are the three main types of cytoskeleton elements: microfilament, then intermediate filament, or microtubules. We have to study microtubules in detail as it covers the major part of the cytoskeleton elements in your syllabus. So now let's begin by each one of them one by one. First we have microfilaments, or also called as actin filaments, as it is made up of actin proteins in it. It is about 7 nanometer in their diameter, and it becomes the most thinnest filament among all of these three. It is a highly conserved cytoskeleton protein that is present at high concentration in nearly all the eukaryotic cells, and also it is one of the most abundant intracellular protein in most of the eukaryotic cells. As you can see in the diagram, this is our G-actin protein. It's a monomeric single unit of protein, or you can say globular actin protein. It is in the heart shape. So when the two monomer of G-actin protein combines together, they form a dimer, and when the three monomer actin proteins combines together, they form a trimer, and so on. The actin monomers keep adding head to tail to form a chain like long structure. When the chain of two G-actin protein comes together, they creates a polymerized double standard helical structure called as F-actin, or the filamentous actin. And at the end, when two chains of F-actin proteins helically coined with each other to form the structure of actin protein. If you look under the microscope, you can clearly see the right-handed helix forming the actin filament, which is similar to the thin rope or filament-like structure. And yeah, as in the diagram, the two parallel F-actin has a distinct structural polarity in them, the plus and the minus ends, which we will see in some other higher classes. Let's see what are the functions of the actin filament. First we have the muscle contraction. When the thin actin filament interacts with the thick myosin filament during the muscle contraction. As you can see in the image, we have muscle, then fibers, and sarcomeres, which is a functional unit of muscle fibers. It has filaments in them, both the actin and myosin filaments. Actin, which is a thin filament composed of polymerized actin microfilaments. You can see in the diagram the thicker line located in the center of the sarcomere. Then we have myosin, which is a thick filament. You can see in the diagram it has small motor proteins on its structure that interacts with the actin during the contraction. You will see these topics in human physiology chapter called locomotion and movement in a more detailed way. For now, you just have to remember that the actin filament plays a huge role in the muscle contraction. Then we have cyclosis or cytoplasmic streaming. It is the directional flow of the cytoplasm within a cell. It helps to distribute the nutrition, organelles, and the other materials evenly throughout the cell. In the plant cells, the cytoplasmic streaming is easily observed under a microscope, where the chloroplast moves around in a circular pattern. This interaction generates a streaming motion of the cytoplasm in the cell. The key function of cyclosis is to ensure the even distribution of nutrients and organelles in the cell, and to facilitate the efficient transport of materials within the large plant cells, where the simple diffusion is too slow to perform. You can clearly see the direction of cytoplasmic streaming is given in the diagram, the red arrows, and the gel or the soul is separated. The gel is the non-moving cytoplasm, or the soul is the streaming cytoplasm, or you can also see the parallel arrangement of actin filaments in it. If you talk about humans, so in the human cells, the cyclosis is much less prominent than the larger plant cells. But a similar process is still observed, they occur to help to move the organelles and the cytoplasmic content in it. No doubt that the movement is slow and less organized than the plant cells, but still the They play a key role in carrying organelles like mitochondria, vesicles, and other cytoplasmic components in it, rather than the visual cytoplasmic flow. Actin filaments are also involved in the cell movement or locomotion and in the shape changing. For example, we have pseudopodia in amoeba. We all know that amoeba is a unicellular organism that moves by the amoeboidal movement, where the cytoplasm continuously flows in the direction of movement by forming the pseudopodia, which is a false feet of the amoeba. So the actin microfilaments in the amoeba polymerizes, or you can say grows at the leading edge of the pseudopodia by pushing the plasma membrane forward by the meiosis motors. Push the cytoplasm along the actin filament by generating the internal cytoplasmic steaming in it. This allows the amoeba to crawl towards the food. And the same process of cyclosis can be seen in the macrophage in humans. Actually, macrophages are the specialized WBC immune cells. It is similar to the amoeba. The macrophages also uses the actin-driven cytoplasmic steaming process to extend their parts of their membrane to the cytoplasmic flow into these extensions helps the macrophage to move towards the infection sites and to engulf the pathogens and the dead cells. Other than these cells, we have few more like fibroblast. We will see this in tissue chapter. So whenever a person get injured, so the fibroblast cell actually helps in healing. What happens is, by the process of cytoplasmic steaming, these fibroblast cells cross there to that cut or injured area and produces matrix to repair that damaged tissue. You will see these cells and these topics in more detail in the chapter called body fluid and circulation. For now, we have to study this much is enough for us. Then we have the another important function of actin filament where the cytokinesis or the furrow formation of the cell takes place. Cytokinesis is the term which means the final stage of the cell division. After mitosis, the nuclear division takes place where the actin microfilaments and the myosin motor proteins assembles together just beneath the plasma membrane at the cell's equator where the furrow formation will form. You can see in the diagram, it generates a force to pull the membrane inward to form the cleavage, or the furrow-like structure forms in the animal human cells to physically divide the cell into two daughter cells, each with its own nucleus and organelles. Fortunately, you will read more about it in the next chapter, cell cycle and cell division. For now, you can remember the actin and myosin both works together to pinch the cell into two cells by the process called cytokinesis. Then we have microtubules. Microtubules has the largest diameter of all the cytoskeletal elements, which is approximately 25 nanometer, whose external diameter is 25 nanometer. However, some books also mention 15 nanometer too, but that is different. Now suppose this is a circle. So this is the outer circumference of the circle, which is 25 nanometer, and this is the inner circumference, which is 15 nanometer, but the 25 nanometer is considered as the standard diameter, which you should remember. That is why it is considered as the most thickest type of filament out of all these three, and it is composed of tubulin protein, which is a form of highly folded globular protein found in all the eukaryote, made up of dimers by the composition of two alpha beta tubulin subunit with each other when they assembles together into a hollow tube-like structure called microtubules. So, when these two globular proteins, alpha and beta tubulin, combines together, they form a dimer. Actually, this dimer is the basic fundamental building block element of the microtubules, and these alpha beta tubulin dimers assembles end to end in a long parallel chain called the protofilament, where the number of alpha and beta tubulin in a protofilament depends on the length of the microtubules, on how long is it. There is no as such fixed number in it. A microfilament comprises of 13 laterally associated protofilaments in it, and then 13 protofilaments align side by side laterally to form a hollow cylindrical like structure of a mature microtubule. And all of these 5 different structures like cilia, flagella, basal body, centriole and spindle fibers all are composed of the microtubules structures only, where you will see cilia, flagella with the 9+2 arrangement in them, and 9+0 arrangement in the basal body and the centriole, where the spindle fiber has no as such arrangement of microtubules in it. You will see all of these in the upcoming slides. You will see a clear or a cross-sectional view of the microtubules in the structure of cilia and flagella in the next slide. So let's begin by their basic structure and functions first. In the prokaryotic cell, we have seen different cell projections like pili, fimbriae and flagella. But the pili and fimbriae are only the prokaryotic character, while the flagella can be in both prokaryote and eukaryotes. And cilia are present only in the eukaryotic organism. So first we have the locomotion in cilia and flagella. They both are the locomotory organs, and we know that. If you see their presence, they are present only in the eukaryotic organism as of their microtubular structure, as I already said in the starting slides. So let's begin by the cilia first. So the cilia is a plural term of cilium. Cilia are the short hair-like outgrowth extending from the surface of a living cell. These small structures work like the oras, causing the movement of either the cell or the surrounding in the fluid. What is ore? Okay, tell me, do you remember your childhood boat rides, or have you ever sat in some small boat or seen someone rowing a boat with the oars? If not, then you must have heard about the Dragon Boat Race Festival, which is held every year in China, where thousands of people rowing a boat all together. Similarly, in the cell, thousands of such oars all working together in a rhythm. That is how the cilia moves. Now imagine the boat is like a tiny organism, or the ore like cilia are the little hair like structure on the cell surface of that organism. Just like the ore pushes water backwards and moves the boat forward, similarly the cilia pushes against the surrounding fluid to move the cell forward. And remember, as the thousands of such ores all working together in a rhythm, that is how cilia moves. In the unicellular organism, cilia are found. We have the example of protozoa like paramecium. You can see in the diagram how numerous cilia is covering its surface from outside. They function as both in the locomotion as well as in the feeding. They help it to move around in water in a coordinated wave-like manner, or also to swell the food particles like the bacteria does. Or in the multicellular organisms, cilia also work inside our body. For example, we have ciliated epithelium, epithelial cells in trachea or bronchi to move dust and mucus out and to trap the microbes upward to keep the airways clean. Cilia in the female reproductive tract, ciliated cells in the inner ear, or the ependymal cells in the brain, or the ciliated cell in PCT of kidney. There are too many such examples are there in our human body. And the number of cilia varies per cell. If we talk about a single ciliated epithelial cell, especially in the respiratory tract, it typically has 200 to 300 motile cilia on its surface, while the protozoa can have even up to 3 to 4,000 cilia on them. That's a big difference, right? That is similar with the other protozoas also, but there are some amoeboidal cells that have no cilia at all. Instead, they rely on some other structure for their movement. But hey, you don't need to go that far for now. You can remember the example of paramecium. That's the classic example given in your NCERT as well. And one thing you should all remember, that cilia causes the movement of either the cell or the surrounding fluid. In the prokaryotic cilia, they are responsible for moving the both the cell and also the surrounding fluids. But the eukaryotic cilia are only responsible to move the fluid, to move the medium around it, not to the cell. Then we have flagella. Flagella is a plural term of the flagellum. It's a long wipe-like structure present on the surface of the cell. And unlike the cilia, flagellas are present in the single cell of both the prokaryotes and eukaryotes. Let me repeat it again. They are present in the single cell of both the prokaryotes and eukaryotes, but not in the multicellular cells. Cilia was also in the multicellular epithelial cells, but the flagella cannot be in the multicellular cells. But don't be confused. Humans are the multicellular organism. They are within the multicellular organism, but only in the single cell, unicellular cells, like the sperm cells. No doubt that these flagellas are comparatively longer than the cilia and are responsible for the cell movement. They are not involved in the movement of surrounding fluid or the medium like the cilia does. They can only move the cell, and also they are limited in number in compared to the cilia. The number of flagella on a prokaryotic cell varies per cell. That depends on the species. We have two different types of flagella. One is the prokaryotic flagella and the eukaryotic flagellas. While the bacterial prokaryotic cells also possess flagella in them, but these are structurally different from that of the eukaryotic flagellas. So the prokaryotic flagellas are made up of the flagellum protein with a minimum diameter of 20 nanometer, that is thinner from the eukaryotic flagella. They are comprises of the simple and the helical structure. If we talk about their number, they can be able to have anywhere around from 1 to several flagellas, typically 1 to 10 or more than that. They have given some specific name according to the number of their arrangement, but that is not given in your NCERT, so we will skip that part for now. Then we have parts of the prokaryotic flagellas, which are filament, hook and the basal body. So let's see what are these. First, the filament is a long, thin and helical structure. It comprises the maximum part of the flagella and it acts like a propeller. It rotates like a propeller, or you can say motor, which is powered by the PMF, protein motive force. It can rotate in both the clockwise or counterclockwise directions. Then we have hook. Hook is again a very short, curved or tubular structure. It acts as the flexible joint that enables the efficient and continuous rotation for the bacterial mortality while connecting the flagella to the basal body, composed of the hook protein, and it is more flexible than the filament itself, which allows it to act as a universal joint. Then we have the basal body. It anchors in the plasma membrane or in the cell wall if there is any, and it acts as a rotatory motor which is powered by the protein motive force PMF for the H plus gradient or the sodium potassium ions across the membrane. This is the basal body is the actual part that spins the filament in either the clockwise or the counterclockwise directions. This rotation moves the bacterium forward and enables it to change its directions as well. They are mainly found in the bacteria and archaeas. Then we have the eukaryotic flagellas made up of the microtubules or the tubulin proteins. It is a very complex structure made up of the microtubule arrangement in a 9+2 pattern, where we have 9 doublet microtubules around the 2 central singlet microtubules with a diameter of 200 nanometers, which make it more thicker than the prokaryotic flagella. Eukaryotic flagellas are found in many of such examples like the protista, animal cells, some plant cells like the sperm cells, and many more. And the eukaryotic flagellas are generally fewer in number, usually 1 or 2 only. They don't have any as such term for eukaryotic flagellas as the prokaryotic flagella does. Like the euglena or the sperm cells has 1 flagella, or the euglenites has 2 flagella, and similar to that. The parts of the eukaryotic flagella are filament, which becomes axome in it, and the basal body. Prokaryotic flagellas don't have hook in their structure as they don't need it. They wipe in a back or forth in a wave-like manner, so they don't need as such hook, or you can clearly see in the diagram, and it arises from the plasma membrane with the structure called the basal body, which is structurally similar to the centriole. And again their ATP is synthesized in their basal body by the dynein arms or the motor proteins. In the eukaryotic flagellas, we have a specific compartment or the specific name for every activity in them that makes it more special. The internal structure of the eukaryotic and the prokaryotic flagella is completely different. The prokaryotic flagella is not given in your NCERT. We have already done about the prokaryotic flagella in the early lectures. So our main focus is on the eukaryotic flagella. But the internal structure of cilia and the eukaryotic flagella are similar. So let's see what are their different parts and their diagrams in the next slide. So first we have the most important part, which is exome or the filament with the 9+2 arrangement in them. Let's break down their this arrangement in an easy or simpler way. The exome is the central feature of the eukaryotic flagella, consisting of the microtubules, where the microtubules runs parallel to their long axial filament. They have some kind of radial arrangement peripheral to the microtubule with the 9+2 arrangement in it. It means it has 9 outer doublet of microtubules on its periphery and 2 central singlet microtubules in its central pair. You can clearly see in the diagram. Look how the 9 pairs of microtubule forms a circular around the periphery, where these 9 doublets are in a pair. We call it the subunits. So we name these subunits as A and B. So the A tubule is complete microtubule with 13 protofilaments, as we know that, and the B tubule is incomplete or the partial microtubule as it has only 10 protofilaments in it. Remember, the B tubule is attached to the A tubule. It is not a complete separate structure in it. Focus on my words and keep your eyes straight on the screen and count it together with me. We have the 9 outer doublets: 1, 2, 3, 4, 5, 6, 7, 8, and this one is 9. And these are the 2 central singlets of microtubule arranged in the center of the exome. Then there is the dynein arm composed of the motor proteins. You can see in the diagram that how they are organized into the rows of outer and the inner arms attached to the doublet of the microtubules within the exome. It has 3 outer arms facing the periphery and 2 inner arms inside the circle, and they are specifically attached to the A tubule of the outer doublet that is fixed. Dynein arms are the ATPase motor protein that converts the chemical energy from ATP into the mechanical force. This released energy helps the dynein arm to walk along the adjacent B tubule by producing sliding between the microtubules. As a result, the coordinated bending of all the microtubules is further transformed into the bending movement of the cilia and flagellas. Dynein arm are the tiny little motor hand, just like the human hands, just like you move your hands to pick up the thing. Similarly, that grabs and pulls on the neighboring microtubules, causing the whole structure to bend and beat and to make the cilia and flagella move in the different directions. Then there is the interdoublet bridge, or also called linkers, in between the dynein arms, which is composed of the nexin protein. That is why it is also called as the nexin linkers. You can see in the diagram that how the peripheral AB doublets are interconnected with the linkers, so they are also called as the AB linkers. So they act like the bridge or the linkers to hold the adjacent doublets together and to stopping their uncontrolled slidings. See, we know that the dynein pulling force gets translated into the bending motion of the entire exome, and this bending is actually what produces the wave-like beating of that cilia and flagella, and the doublets cannot freely slide, all thanks to the nexin linkers that prevents excess sliding by converting sliding into the bending. You can remember nexin linkers like a rubber band. It stretches and recoils to give stability and flexibility to to the exome. Then we have the another bridge called the intersinglet bridge. As in the 9+2 exome, the central +2 pair of singlet microtubule is referred as the S1 and S2, or you can also say C1 and C2, which means singlet 1 and the singlet 2. That is not just a freely floating singlets, but these two singlets are connected to each other by the fine protein lines called the intersinglet bridge. It stabilizes the central pair to keep these two singlet microtubules correctly aligned in their position. And most importantly, it maintains the symmetry of that circumference by preventing the central pair from rotating independently and ensures their proper wave propagation of the exome. Then there is another T-shaped protein complexes which are called as radial spokes. Radial spoke got its names from the radii of the circle. They are similar to that radius in a circle which connects the circumference, the outer ring, to the center. Similarly, these are the structures extend from the outer doublet towards the central pair of the microtubules. Or you can also compare it with the central spoke of the bike wheel or the cycle tire, where each radial spoke has three distinct parts in them. One is the base, then the stalk, and the head. So the base is anchored from the A tubule of an outer doublet, and the stalk is the thin protein rod like that extending inwards towards the central pair. Then at the end it has head. Head is the broad flat region at the inner end of the stalk which lies perpendicular to it and faces the central pair of the singlet microtubule C1 and C2. The head of the radial spoke is not spherical or round like the human head, but it is flat and horizontal. So because of their vertical long stalk and the horizontal broad head, the whole structure looks like the T-shaped while looking in the cross-sectional view under the electron microscope. If we look on their arrangement, so suppose if it has one radial spoke per outer doublet, then we have the 9 outer doublets, so it must be the total 9 radial spokes attached to the each doublet, and it also performs some important functions like in the signal regulation, as it acts as the sensor to transmit the signals between the central pair of complexes and the dynein arm of the outer doublets to ensure that all the dynein doesn't pull at once, and also to maintain their coordinated beating. As they are in between, so they are also helps to maintain the space between the outer doublet and the central pair. What if we remove all the radial spokes from the exome? See, we know that these spokes connect the rim. Rim is in the tire, you can assume with it. So the outer doublet to the central pair. So if you remove all the spokes, the rim can still spin, but it becomes very unstable and uncontrolled. They can disrupt the regulation of the dynein arms, and without the radial spokes, the exome cannot bend properly, and the cilia and flagella will completely lose its functionality, or they can even be immotile and can be paralyzed also. So this is again the same diagram of the cross-sectional view of the internal structure of cilia and flagella in a more magnified way, even though the microscopic view of their cross-sectional structure is also attached on the left side of the screen for your conventional clarity. So you can check this out. Then we have central sheath. It is sorry for my bad thought. So then we have the central sheath. It is in the red color in the exome. You can see it may looks like a membrane, but actually it is not. It's a proteinaceous sheath which surrounds the central pair of the singlet to hold the central singlet in their place. And you can imagine this sheath, this cover as like an insulating sleeve around the wires. As it is in red color, you can imagine it better. So it keeps how in the wire it keeps them stable and helps to manage the flow of the signal or charges in it to rest of the structure. Similarly, it works with the radial spoke to transmit their signals from the central pair to the outwards to the dynein arm. By this ensures that the dyneins are activated in a coordinated sequence, so that not all dyneins are activated at once. This also helps to determine the direction and pattern of the cilia and flagella beating. Without the central sheath or radial spoke, they both are important. Like without them, their interaction, the complete movement become uncoordinated and paralyzed. So together the central sheath and central cingulate microtubule together they can form the central apparatus, and this complete highly organized exome structure is covered by the plasma membrane. You can see. So yeah, this is all about it, the internal structure of cilia and flagella. Now we'll move by the centrioles and the basal bodies. So the basal body is at the base of the structure of cilia and flagella, which serves as the foundation or the base of their structure, which acts as a gate for the entering of proteins into their structure of cilia and flagella. Both the cilia and flagella emerge from the centriole-like structure called the basal body, which is again composed of the microtubules with the 9+0 arrangement in them. 9 periphery triplets with the 0 singlets in them. Their function is to act as MTOC, microtubule organizing center, and it also anchors the flagellum to the cell and acts as a template for the microtubule assembly. The electron microscopic study of the cilia and flagella shows that they are covered with the plasma membrane on its outer side, which makes it a true extension of the cell. But the cell wall is absent on the exome. It is present on the rest of the part of the cell but not on the exome. You can see in the diagram. I have attached this external structure of the cilia and flagella, where you can clearly see the centriole-like structure of basal body is also attached on the base of the exome. So both the structures of the basal body and the centriole are structurally similar to each other. They both are composed of the 9 triplet microtubules in them. So let's do their structure together in the next slide. So before going to the structure of centrioles, let's do a quick brief comparison between the structure of cilia and flagella. So what we saw is the cilia is shorter in size, hair-like with the hair-like appendage from the surface of the living cell, which is about 5 to 10 micrometer in their size, while the flagella are long and more thread-like appendage of the cell surface, which are up to 150 micrometer in their size. Their Latin words are also written there, and their cilium is the singular of cilia and the flagellum is the singular of flagella. While the cilia are in the eukaryotic cells, but the flagellas can be in both the eukaryotes and prokaryotes. But we have done about the eukaryotic flagellas in detail. And you can also remember the number of cilia per cell is very high, but the flagellas are few in their number. And you can remember their beating pattern, like the cilia beats in a coordinated manner in a oar-like movement, but the flagellas beats independently in undulational movement. Cilia can move both the cell and the medium, while the flagella can move only the cell. Don't confuse that the cilia take part in the locomotion, attachment and feeding and sensation and all of these, but the flagellas are only involved in the locomotion. So yeah, that is it for today. Thank you for being till the end of this lecture. In the next part, we'll finish the centrioles. Till then, keep learning, keep smiling. Have a good day.